when a photon of light strikes a pigment molecule in the light harvesting complex on the thylakoid membrane, which is in the chloroplast. The energy is transferred to other pigment molecules via resonance energy transfer until it reaches one of the two special chlorophyll A's in the reaction center of photosystem 2, called P680. It is important to note that only energy is transferred and not the electron itself. This excites one of the electrons in P680, raising it to a higher energy state. When the electron in P680 is photoexcited, it is captured by the primary electron acceptor in the reaction center of photosystem 2. At this point, P680 is missing an electron, creating an electron deficit. To replenish this, a molecule of water undergoes photolysis and is split by an enzyme into 2 hydrogen ions, 2 electrons, and 1 oxygen atom. This oxygen atom will immediately combine with another oxygen atom to form O2, which is released as a byproduct. The electrons from water will then replenish the electron deficit in P680. Now that the primary electron acceptor has a photoexcited electron, it is passed from photosystem 2 to photosystem 1. It is worth noting that PS2 and PS1 are named in order of when they were discovered, so PS2 is actually the first system in a non-cyclic reaction, despite its naming. The electron travels through the first electron transport chain, consisting of plastoquinone, the cytochrome Bf complex, and plastocyanin. These electron carriers are arranged in increasing electron affinity, ensuring that the electron moves unidirectionally. The electron will move through a series of redox reactions, that is oxidation and reduction reactions, where in each step it is dropped to a lower energy level, releasing free energy. The free energy is used to pump protons against their concentration gradient, from the stroma into the thylakoid lumen. The proton gradient is important for chemiosmosis, which is the process where energy stored in the proton gradient across a membrane is used to drive synthesis of ATP, in this case through photophosphorylation. The proton gradient is generated through 2 steps, one being the photolysis of water, which releases 2 hydrogen ions. Second, the cytochrome Bf complex pumps the H+ against the H+ concentration gradient. Before the electron can be transferred from plastocyanin to PS1, a photon of light will strike the pigment molecule in the light harvesting complex of PS1. Via resonance energy transfer, the energy is transferred to one of the 2 special chlorophyll A's in PS1, known as P700. The photoexcited electron of P700 is captured by the primary electron acceptor of PS1, creating an electron deficit. This electron deficit of PS1 is then replenished by the electron that came from PS2. Now that the electron deficit in P700 is replenished, the newly photoexcited electron from PS1 will move down the second electron transport chain, where it is transferred to an electron carrier called ferredoxin. From there, the electron is transferred to an enzyme called NADP reductase, which reduces NADP+ to NADPH by adding 2 electrons and 1 proton. In a non-cyclic light-dependent reaction, NADP+ becomes the final electron acceptor, creating NADPH, which is used in the next stage of photosynthesis, also the Kelvin cycle. And that concludes our look at the non-cyclic light-dependent reactions. I hope you found this video helpful, and if you're interested in learning more about photosynthesis, such as the Kelvin cycle, be sure to check out my upcoming videos.